once you have a specimen that you want to image in the microscope, you're almost always going to have to do something to enhance the contrast in that image. Contrast being the amount of bright and dark in the photograph. If we look at the image on the left on the screen here, number A, and this is from your textbook, this is some cells on a plate, and you notice it doesn't look like there's a whole lot there, and that's because human tissue is by and large clear and colorless. So we have to do something to impart contrast to let us see the variation in the structures inside our cells. So on this page, we're viewing two different ways of imparting contrast using just light itself. So panel B over here is an example of a phase contrast micrograph. So let's see if I can write on here. I might have to animate. I don't know. Phase contrast. There's supposed to be an R right here. Very fine. Okay, so in phase contrast, what we're going to do is, without getting too technical, we're going to play with the light such that very small changes in density, so even very fine structures, are going to be rendered out as big differences in optical contrast. So for example, if we look at this nucleus right here, so this nucleus right here in this cell, we can see the nucleus itself appears darker than the cytoplasm, and indeed we can see some of the chromatin and the nucleolus inside of it. That nucleus is present in the first image. We can see it right here. However, there's not enough natural contrast in here. So by essentially allowing changes in shift that occur in the light as it passes through, so for example right in here, okay, the light was slowed down just a little bit because this tissue is a little bit more dense than the tissue around it. And so in phase contrast, we take that shift in phase, right? The light waves are actually slowing down, and we amplify that shift in phase as a visual shift in contrast. A related method called differential interference contrast, or D, that's supposed to be a D-I-C. So, let's see here. Let's just fix that real quick. So differential interference contrast, or DIC, that's more like a D, close enough, is even more complicated than phase. But what it's going to do is, again, we're going to monkey with the light. In this case, we're going to shear it using some polarizers. We're going to pass that light through a couple of prisms, and we're going to repolarize it as it comes back out. And essentially what that's going to do is give us this kind of three-dimensional appearance where small differences in density are once again rendered out as differences in contrast. So both phase contrast and DIC are methods by which we can play with white light and impart contrast. And this doesn't necessarily require you to fix your cells. These cells are probably live cells. So these definitely have some advantages. However, most of the time, it's okay to kill the cells we're going to look at. So normally, what we're going to use is some method of staining. Now, as we go through the semester, we'll encounter several different ways that tissue can be stained. But the most common method of tissue staining is called H and E. And the H stands for hematoxylin. It is a blue basic dye, and the E stands for eosin, a red acidic dye. And because opposites attract, the basic dye will bind to things that are acidic, and the acidic dye will bind to things that are basic. So again, this is another picture from your textbook, but if you look in the bottom right-hand corner, all of the kind of bluish purple dots, those are nuclei. Nuclei contain deoxyribonucleic acid. So since nuclei tend to be acidic, they're going to preferentially bind the more basic dye and appear blue or purple. Whereas collagen protein, which is this pink wavy substance in this bottom right picture, collagen tends to be kind of basic. So it likes to bind the red acidic dye, eosin. And there are numerous combinations of dyes that bind depending on the pH of the tissue. There are dyes that bind metals preferentially. there are tons and tons and tons of different dyeing regimes you can use to image different substances. H&E is the most common. The other one we're going to see a lot is the trichrome stain, which uses 3 different dyes to impart even additional contrast. So one way or the other, if you're going to look at something that came from a human being in the microscope, you're going to have to use either an optical contrast technique like phase contrast or DIC, or you're going to have to stain the tissue using H&E, trichrome, or some other staining regime in order to impart contrast on your tissue so that you can differentiate between the various components.